A leukemia patient undergoing chemotherapy suddenly crashes into acute renal failure from tumor lysis syndrome. Yet simultaneously, the laboratory prints a uric acid level that appears within the normal reference range. This bedside reality completely contradicts the automated test. To understand why the laboratory results can be so deceptively clean during a metabolic crisis, we have to look at the unique history of the molecule itself. In human architecture, uric acid functions as an active metabolic signal that connects dietary intake to the global redox security of our extracellular fluid. This system began during the Miocene epoch, when a nonsense mutation, a genetic typo that creates a premature stop sign, permanently silenced the UOX gene in our hominoid ancestors. This mutation stripped us of the enzyme uricase, the chemical scissors needed to break down insoluble uric acid into harmless allantoin. As a result, human serum urate concentrations run 10 times higher than almost all other mammals. The architecture explains the danger. Deprotonating the acidic nitrogen protons at the N9 and N3 positions creates a negative charge, attracting sodium to form a monosodium urate complex. Its anodiol-like structure acts as an electron sponge, neutralizing unstable free radicals, preventing vascular rusting. This single molecule provides 50 to 60% of the total antioxidant scavenging capacity in human blood plasma, supporting endothelial health and extending our species' lifespan. But we maintain this defense at a steep physical cost. The human body operates perilously close to the molecule's strict solubility threshold of 6.8 milligrams per deciliter. By selecting for this mutation, evolution prioritized a potent antioxidant shield over physical solubility, creating a system that protects the vasculature but fails the moment it is pushed past its physical limits. Managing this precarious balance falls to the kidneys, which act as master inventory controllers for a circulating urate pool of roughly 1.2 grams. The kidneys use a 4-phase sorting matrix. First, the glomerulus filters all urate into the waste tubule. Immediately, Urate-1 transporters reabsorb 98% back into the blood. Next, pumps secrete half back out. Finally, Glut-9 salvages the rest, excreting only 10%. This energy-intensive multi-gate system gives the body precise control, allowing the kidneys to adjust urate levels minute to minute based on hydration and oxidative stress. These baseline levels also shift with human hormones. Estrogen directly downregulates the Urate-1 transporter in the kidney. This hormonal signal tells the kidney to leave more urate in the waste fluid, which is why premenopausal women maintain lower circulating levels and are largely protected from gout. The baseline drops even further during the first trimester of pregnancy, as blood volume expands and the kidneys enter a state of gestational hyperfiltration. Because the human architecture manages such a strictly low baseline during pregnancy, a sudden third trimester urate spike is a critical early warning sign for preeclampsia and endothelial ischemia. When serum uric acid exceeds 6.8 milligrams per deciliter, physics overrides biology. The dissolved urate physically drops out of the blood. This crystallization targets the peripheral joints, such as the big toe, through a localized temperature trap. Joints in the extremities are naturally cooler than your core body temperature. If your foot gets cold at night, that drop in temperature physically lowers the solubility threshold of the surrounding fluid. The dissolved urate shifts almost instantly into highly sharp, extremely jagged, needle-like monosodium urate crystals right on the surface of the articular cartilage. Patrolling macrophages encounter these crystals and recognize them as a danger-associated molecular pattern, or DAMP. Because the crystals are physically sharp, they puncture the macrophage's internal sacs, triggering the NLRP3 inflammasome and releasing an interleukin-1 beta cytokine storm that floods the joint with white blood cells. While cold toes cause localized gout, systemic overloads are often driven by hijackers like high fructose corn syrup. Fructose metabolism burns through cellular energy so fast, it strips ATP down to its raw core, AMP. This adenosine core is a purine base that floods the catabolic pathway, forcing the enzyme xanthine oxidase to churn out a massive surge of uric acid. In contrast, patients with hereditary Urate-1 mutations waste all their urate. Lacking your primary electron sponge, they suffer massive oxidative damage and exercise-induced acute kidney injury. Whether the inventory is overloaded by dietary ATP depletion or stripped bare by genetic wasting, mismanaging the urate balance destroys either the joint architecture or renal integrity. This complexity explains the clinical paradox, how a tumor lysis patient can have a massive uric acid spike that the automated laboratory simply fails to see. The gold standard automated assay uses a uricase enzyme to oxidize the uric acid in a blood sample, which generates hydrogen peroxide. Then a peroxidase enzyme turns that hydrogen peroxide into a vibrant dye. The machine measures the color intensity to calculate the urate level. This process is vulnerable to chemical interferences, specifically from high doses of vitamin C. Ascorbic acid is a powerful reducing agent. It outcompetes the dye, neutralizing the hydrogen peroxide before the color can develop. The machine scans a clear tube and reports a falsely depressed uric acid level, masking a severe hyperuricemic emergency. But the tumor lysis patient falls into a second trap, the ex vivo oxidation artifact caused by the drug rasburicase. Rasburicase is a recombinant version of that ancient uricase enzyme. It is injected into the blood to instantly cut insoluble urate into allantoin, saving the kidneys. However, the drug continues working inside the blood draw tube if it is left at room temperature. It literally devours all the uric acid inside the sample before it ever reaches the laboratory analyzer machine. Blind reliance on automated results without correcting for chemical reduction or drug kinetics leads pathologists to interpret a cleared test tube as a cured patient, risking fatal renal occlusion. Managing these traps requires strict diagnostic stewardship and adherence to timing protocols. The Choosing Wisely clinical guidelines explicitly state that you should never test a patient's uric acid levels during an active acute gout flare. During a flare, the body's chemistry is in chaos. Massive amounts of urate have shifted out of the blood and into the tissue, making the lab reading appear paradoxically normal or even low. Clinicians must wait 2 weeks after the flare resolves to get an accurate steady-state baseline to base their treatments on. Failing to wait leads to the treatment paradox known as the asymptomatic hyperuricemia trap. A doctor sees a high uric acid level on a routine checkup and immediately prescribes the drug allopurinol to suppress production. Artificially crashing blood urate levels rapidly alters the body's equilibrium, which causes microscopic background crystals to start dissolving. As these background deposits dissolve, they shed their protective protein coatings, exposing raw, jagged crystalline needles to the local immune cells. The medication prescribed to fix a spreadsheet number actually triggers an agonizing gout flare in a patient who previously felt fine. Uric acid is an active evolutionary defense. Managing it requires treating the complex human architecture of the patient rather than fighting to zero out a single laboratory metric.